All right. Hi everyone. Thank you guys all so much for joining us today. We collectively are the BioImaging North America Early Career Working Group, and we decided that we wanted to do this BioImaging ToolBox workshop where we're just going through and showing everyone a few of our favorite things that we like to use for helping us design and execute and analyze an entire imaging experiment. For those of you that are just joining, we have some QR codes at the top of our title page. We have a survey link that we would love for you all to fill out just to let us know what tools that we're covering today that you might want to learn more about, or if there's something we didn't cover today that you would like to learn more about, there's an option for that, as well as any questions you might have or general feedback. We also have a resource document that we put together that has a list of organizations as well as learning material for the different tools that we covered today. And Mike Nelson, who is one of the people heavily involved in the Image SC forum. He put together a forum post so that way, if you have any questions that you would like to have answered, you can feel free to go over there, post them. You're also welcome to put them in the chat or that survey. Eventually, all of those questions and answers will be migrated to that forum post for just continual reference for everyone. All right, and with that, we're going to get started. So today we have a pretty extensive speaker lineup, including myself as Kristen, as well as all of these other lovely people that you see the videos on of. The majority of us are part of the early career working group, but a few of us are outside members that we invited in just to give some quick talks about some of the tools that they really enjoy working with. All right, so why did we decide to put something like this together? Imaging experiments are really complicated to do, and for those of us that have, even many years of experience, it can still take quite a bit of planning in order to conduct one properly. And there's a lot of tools out there and a lot to sort through. So, one of our things that we're hoping to get across today are some of the tools that we think are reliable. Unfortunately, a lot of tools over the years might not be supported, and sometimes it can be difficult to figure out which ones those are. And we also want to show you guys how you can be fully supported through the duration of an imaging experiment from design and acquisition all the way through your analysis. And the following presentation are just some of our favorite tools for getting the job done. All right. So if you would like to uh, fill out a quick survey. This is just another plug for it. We have a QR code here. We'll also be putting that link in the chat periodically. Let us know what you liked, what you'd like to learn more about, and we can try and plan those kinds of talks in the future. Right. So, what makes a good imaging experiment? We have this large pipeline that starts with what I am lovingly referring to as pre-analysis, which is all of the work and the planning that you need to do before you really start to sit down at the microscope and imaging your samples. We have a lot of tools that we'll be chatting about today. That is what's shown on this screen is just a little icons for all of the tools we're covering. We also have, your imaging portion. There's a few things that go along with those tools that you can use to help that process be a little bit more smooth. Um, Once you have your data, how do you process and analyze it? There's a lot of tools out there. We're covering some of our favorite open source ones. And then if you get stuck, where do you go for help? So we have a few forums that we really enjoy reaching out to that we're going to go over as well. All right, so we're going to start off with that pre-analysis. This is the work that you need to do before your experiment. And this will be the last time I'm talking until a little bit closer to the end. But we wanted to walk you through how do you figure out what kinds of fluorescent proteins or fluorophores would work well for your microscope to use for your imaging experiment. Fluorescent Protein Base, or FP Base, is an online open source community editable data set of fluorescent proteins. And their primary objectives are to help aggregate structured and searchable fluorescent protein data that is of interest to the imaging community and fluorescent protein developers. It was developed and designed by this single guy, Tally Lambert, at Harvard Medical School back in 2018. And honestly, this software or this website has like really flourished since then. It's a really great resource. So what can you use it for? You can explore the lineage and the directed evolution of fluorescent proteins, which I think is kind of cool to see. You can see relationships between different fluorescent protein properties, compare spectras with common filters and light sources. You can calculate FRET relationships if that's of interest to you. You can also create and share your own curated protein collections if you wanted to. So this would be like your own private little, not private, but like your own little curated resource for yourself. You can also build a spectra viewer that's customized to the specifications of the microscope you use, so you can very easily figure out what fluorescent proteins or dyes might work with your microscopes. So just a quick little demo of what you might see when you look at a fluorescent protein on the on fluorescent protein base. This is for GFP, where we had the spectra at the very top. And I'm just scrolling through and showing you quickly like some of the different information you can learn about, I'll just play it one more time, about a particular fluorescent protein. So we have emission spectra, brightness, other information about the protein, as well as resources you could look at. In terms of looking at property relationships, you can look at an interactive chart that shows the emission and excitation wavelength properties of different fluorescent proteins. They have filters on this particular tool that allow you to very quickly find fluorescent proteins that might work well for your emission and excitation properties of your microscope. You can view spectra of your fluorescent proteins. So being able to know what those are allows you to make informed decisions about what kind of fluorescent proteins you're going to use. You can view multiple fluors at the same time with this viewer. And like I mentioned earlier, you can use these spectra with your microscope setting to be able to see, will this protein work with my excitation and emission filter properties. So this is just a few examples of looking at green fluorescent protein and how it might be excited and collected by different excitation and emission filters and lasers with this particular microscope setup. So some quick pros and cons. You can easily find multiple options for similar fluorescent proteins. You can create accessible and shareable libraries of proteins. It's very community driven, which I absolutely love. Some of the cons of it is that the microscope viewer unfortunately cannot view multiple fluors and light sources at the same time. So that is a downside, but the spectral viewer is able to view multiple spectra or fluorescent protein spectra at the same time. And because it does rely on the community to contribute to this, there's no one actively working on the back end that's getting paid to like support this website. So it is both a pro, but also a con in that it takes us to make this a better resource. Right. And with that, I'm going to hand it over to Natalie, who's going to walk you guys through one of her favorite tools. Oh, I'm sorry. Can you request control again?
It says I have control. Or maybe not anymore. A second. Rest. All right. Well, hi, everybody. Thank you all for being here. And thank you, Kristen, for introducing FP-Base to us. Today, I'll be talking about BenchSci, or specifically Ascend. Let me go back. Oh. Okay. So Ascend, or BenchSci, is an AI-powered online platform that will curate published data so that researchers can find validated antibodies and reagents for their experiments to use. Their focus is really to improve preclinical research efficiency and reproducibility. And it began as a startup from the University of Toronto that was launched in 2017 by 4 founders, Tom Leung, David Chen, Elvis Rionda, and Liron Belidzin. So some key features the reasons that I like using it is because it's a very easy to use interface and it's simple to navigate. You can search by a protein target, the tissue that you're using, the assay type or the application, specific antibody specifications, and even add like supplier filters to see who you can order from for those products. And then you get access to experimental context from peer-reviewed publications, as well as the direct links to the products of interest themselves. So here is a video just kind of showing how it works. So on the website, you can look up the protein or gene target. You look up your application, so immunofluorescence in this case. You specify what tissue type you're going to be using it on. And then you can scroll through just the figures that people have published using this antibody or reagent. You can look through a lot of the different types of publications, the product info, as well as direct links, like I mentioned. So there are obviously pros and cons to something like this. Pros was it really does save time on literature review. If you're looking for a specific protein or antibody or reagent, then you can kind of scroll through after filtering it all out and find what research studies have been peer-reviewed that utilize this antibody. It helps to reduce trial and error with reagent selection because you're using stuff that people have validated for you already. It enhances reproducibility through evidence-based choices. However, there are cons. So some reagent data may still be missing or limited because it only was started in 2017 and may not cover all experimental variations. So there's a lot of different applications that people have used it for, but there are limitations to maybe some better for Western blot or for immunofluorescence, but the data is definitely not fully there yet. And it is best suited for institutions with journal subscriptions because they give you a link to the peer-reviewed paper, but then you still have to have access to that paper to look through the supplemental data and stuff like that. So with that, that's pretty much just one thing that I like to use, so BenchSci Ascend. However, there are all alternatives, and I'll leave Andy to tell you about an alternative to BenchSci.
Thanks, Natalie. So yeah, a little bit of deja vu on this one, but you can basically see that we're going to talk about a very similar product here. So another website, this is Site AB. It was an academic spin out from a group that was just interested in trying to, as Natalie suggested, kind of improve the literature review and reduce the kind of trial and error aspect of picking antibodies. I'm sure a lot of people in the audience have had that pain of, buying antibodies and then finding that they don't actually work, or they work better from one company than another, that sort of thing. So this permits, like Ascend, searching for antibodies by clone or target in order to identify those that have been published, and it ranks them by the number of citations. So you can essentially filter antibodies based purely on the number of times they've been published. And then it links to those publications and often details the techniques and dilutions. And importantly, for the context I was considering when I used this, was species reactivity. So when I click through here, so it's just going to play a short video of this function for a target that I was interested in. Just going to wait for that. Okay. So we're going to search for vascular endothelial cadherin. So you can search, find all of the VE cadherin antibodies, and you can see here we have one that is a rabbit host. We have a different one that is a mouse host. And they both have cross-reactivity against human and mouse. And you can see some example images from Western blot. You can scroll down and see what types of techniques these have been used in, how many publications have been published for things like immunofluorescence, immunocytochemistry, Western blot. You can, again, link, like in Ascend, to the images. So you can actually do your own essential review of the images in the various tissues that people have used. So you can potentially double check that whatever has been published is kind of targeting the area that you want, and you can go directly to the publications themselves. It also provides useful information sometimes in terms of dilutions that might be helpful, and allows you to filter based upon which species you're trying to target. Another feature that I really like is if you have a clone, you can search for the clone, and sometimes you'll find that the same antibody is listed from multiple companies. And so you can often save yourself a little bit of money by finding the same clone of antibody targeting the same target from a different company. So for instance, in this panel, I really wanted a goat antibody that was going to target for whatever reason. Probably I'm building an IF panel that uses secondaries, and I can see that I wanted it to target mouse. So I have a goat anti-mouse VE cadherin antibody. Check the images, it looks good. So I can know with relative confidence it's not foolproof, but 225 citations implies that there's been at least a good number of scientists who have tested this out before me. So I'm just going to click through. So much like you have with Ascend, there are some pros. Obviously, it saves time in the literature review. Hopefully, it does reduce that trial and error with reagent selection. If you pick an antibody with 1,000 citations versus picking an antibody with 1 citation, you can have a little bit more confidence in the 1,000. It enhances the reproducibility through evidence-based choices. And hopefully, you can find that same clone or conjugate cheaper elsewhere. Some other cons, some reagent data is definitely missing or limited. I would be very careful with species reactivity. In that first initial headline, it always says, someone has shown cross-reactivity with human, and then when you look at the data, you might find that that was only 1 citation. So you may need to just double check that actually, it's still showing you what you're looking for when you're searching it. I would actually recommend using this in tandem with something like Ascend. Hopefully these databases collectively can cover a lot more. So it's potentially a technique to use together. And also techniques overlap and names overlap. Sometimes you have to be quite specific with the spelling. So search for VE cadherin, vascular endothelial cadherin, VE cad, variations of the same name. And then techniques overlap. You'll often be looking for immunofluorescence. Maybe you actually want immunocytochemistry or or cell immunofluorescence, paraffin immunofluorescence. It's worth looking at a bunch of these different options in the drop-down lists to make sure that there is some crossover there. So that's Site AB, and I will pass over now to the imaging section.
Great. Thanks, Andy. Yeah. Hi, everyone. My name is Quinley, and I'll talk about the Nyquist calculator, which is used to determine optimal XYZ sampling to improve your image resolution. While there are several options available online for calculating Nyquist, the most famous Nyquist calculator is the one provided by Scientific Volume Imaging. This is a company founded by Dr. Hans van der Voort out of his work at the University of Amsterdam. And SVI is best known for their Huygens software, which is an image processing software you might have heard of before. While Huygens does require a license to use, SVI's Nyquist calculator is free. Here we can see a diagram of cells on the right, and we can see the impact that pixel size has on the ability to distinguish objects and resolve details. When thinking about resolution and sampling, there are some important concepts to keep in mind. One is Rayleigh's criterion, in which the minimum resolvable distance between 2 points in a diffraction-limited optical system can be calculated using the wavelength and the numerical aperture of the objective. And per Nyquist's theorem, it's recommended generally to sample at least 2.3 times the highest frequency to faithfully reproduce structures at the maximum resolution allowed by the objective. So putting these two concepts together, we can generally aim for your pixel size to be about 2 to 3 times smaller than your theoretical optical resolution to be sampled according to Nyquist. Okay. So here we have a general walkthrough of the Nyquist calculator. First and foremost, it's important to know your microscope system and optical configuration. First off, what kind of imaging are you doing? Confocal or wide field or light sheet? What's your numerical aperture, your excitation emission wavelengths, and the type of immersion that you're using and its refractive index. If you're interested in generating a point spread function based on this Nyquist sampling, you can click the checkbox and fill out a few more details. What's especially useful about this calculator is that for any parameter that you're not sure about, you can click on it and it'll give you a thorough explanation on the concept and how to determine the number to put in the calculator. So when you're all done, you can click calculate and then you'll get your optimal XYZ sampling. Okay. All right. Okay, so some pros and cons. So this Nyquist calculator will help improve your image resolution, and this is especially important when doing deconvolution, but there are some things to keep in mind. Just be aware that the SVI calculator does go beyond the 2x minimum recommendation for Nyquist, which may not be practical for your needs. And related to that, it can sometimes be a practical challenge to achieve perfect Nyquist, especially when using low magnification objectives. And although Nyquist is an ideal standard, there are some use cases to undersample. For example, in live imaging, where you might bin your image to capture more light. For some alternatives, check out our resource doc for a couple other alternative calculators, such as Tally Lambert's iScope calculator and Nikon's resolution calculator. And just note that some vendor instruments like Nikon and Zeiss do have software features that lets you achieve Nyquist with just the push of a button, which you can see here. But keep in mind how these different options are satisfying the Nyquist criterion, as some might have your pixel size to be two times smaller, while others can even go down to 4 times smaller. Yep, and so that's everything. Thanks so much.
Okay, thank you, Quinn. There we go. So Micromanager is the tool I'm going to talk about, which is an open source software package that actually controls microscope hardware and cameras and acquires images. So it's a sort of an academic and open source alternative to the software that a microscope vendor might ship with a microscope. It was started in Ron Vale's lab then at UCSF exactly 20 years ago by Nico Sturman and Nenad Amodai, and has since had many contributors, both from academia, but also from companies that manufacture microscope equipment. And I've been involved in its development for a little over a decade as well. So as I said, Micromanager's role is to control the microscope hardware, and this can be anything from the main microscope body, the stands, and cameras to XY stages, focus drives, light sources, filter wheels, and many other things. They could be incubators, for example, all sorts of things that go on a microscope can be controlled. And this applies to both commercial and home-built microscopes. So if you have a microscope that is built out of parts from multiple companies, this is one tool that can drive those microscopes. And the main thing that we do is it runs experiments, of course, by automating time series acquisitions, multi-stage position, or in some cases, multi-well plate imaging, Z-stacks, and multi-channel imaging. And also, Micromanager supports scripting and programming. And so people have built plugins for Micromanager to do special things based on that. And also we have a Python scripting interface whose development is led by Tally Lambert, whose name seems to come up a lot today. So this, because we control so many different pieces of hardware, we have a built-in configuration interface, and it will allow you to add devices. Currently, we have over 200 different types of devices that we support. And if you want to learn what devices are actually supported, you can go to the devices section on the website. This is what the main window looks like. It has on the left the sort of the basic actions: snap an image, view live images from the camera, go to multidimensional acquisition, which I'll show next, and various controls for the devices. And on the right-hand side, we have these user-created settings that will control multiple things on a microscope in a consorted way. So this is used to, for example, specify the channel, which might be a combination of a laser line or other illumination line, plus, for example, a filter turret position. Now, the main experimental interface is called multidimensional acquisition, which is shown on the right here. And this allows you to specify in a fairly simple interface a time series experiment, stage positions, and I don't show it here, but you can do things like create a grid or a multi-well plate position list, and then through focus stacks, and then channels. And we also support software autofocus and some hardware focus maintenance devices used in conjunction with this. And from this, you obviously can acquire images. And what's shown here is these sort of sample images that are displayed by the software demo devices that we include. And we have nice histograms, which are critical for acquiring usable images at the microscope. Just quickly, some pros and cons that I can think of. I think one of the strengths of Micromanager being an open platform where people can contribute is that it supports a very wide array of devices, including from different vendors. And I guess one of the cons is that it's not tailored to any particular microscope system. So it's not a turnkey experience like you would get from the software that's bundled with a microscope system sold by a large company. And I guess one thing that's a frequently asked question is that we do not, for the most part, support scanning microscopes. So we do spinning disc confocals, but not laser scanning, except for some home-built examples. And I just listed a few other open source tools that are in the same category: ImSwitch, the Python microscopy environment, Navigate, and ACQ4, which if you're interested in this kind of thing, you might want to look at. And with that, I'm going to turn it over to the processing and analysis section.
All right, so I'm going to take it over real quick just for the first portion of this. So once you get your data, we got to process and analyze it and learn, what's really interesting about our data. So I'm going to talk to you guys about QuPath, which is one of my favorite open source tools to use, as well as to teach other people how to use. It was created by Peter Bankhead as a solution that he came up with for an image analysis problem he was experiencing. It is open source, community driven, and it is pretty user friendly as well as scalable. It's really amazing for analyzing those large pyramidal images. So these are images that you might get off of a slide scanner where you have multiple layers of resolution. They're very, very large tissue samples. It's really great at handling those. It's excellent for analyzing 2D images in general. It's made for easy batch analysis. It also has several built-in machine learning tools, as well as a lot of machine learning AI plugin tools available, such as CellPose, Stardust, SAM, and as of QuPath 6.0 Instance Egg. So I'm going to walk you guys through just it's a very fast-paced demo, but just to show you guys a few ways that you can use QuPath. So we have an image from a confocal microscope. And what I'm showing you guys here very quickly is using the built-in cell detection modules where you can select the channel of interest. In this case, we're focusing on nuclei. And in this next example, we're using the CellPose plugin to detect those same nuclei. And hopefully you guys have noticed that there are slightly different results when we're using different tools. We can also collect measurements and visualize the differences of those measurements very easily. In this case example here, I'm now showing using that same cell pose plugin to detect both the nuclei as well as the cell outline of all of the cells within the image. We can collect those measurements in all of the cell compartments and export them as CSV and TSV files. We can also classify our objects based on the measurements that we're collecting from the data. So if you have multiple classes of cells that you want to be able to identify and label, you can do and accomplish tasks like that. We can also use simple thresholding techniques to be able to classify cells as well. And that's some of the examples that I'm showing here is we're using measurements that are collected in the cytoplasm or in the nuclear compartment of the cells and using those to classify them. There's also built-in pixel classifier tools where you can use those to segment the things that you care about. So here I am drawing lines to indicate to the software what is a cell and what is background. That's the difference between the gold lines and the gray lines. And once I've gone through and annotated several examples of those, I can see a preview and then go in and fine-tune those results as needed to get as accurate as of a result as I need to. So I'm just finding a few patches here and there to fine tune what is background or what is foreground, in this case, the actual cells. From there, you can make an actual object. So in this case, from this preview, I'm just generating an object that is all of the cells, not individual objects. You can do something similar, but in this case, we're detecting the nuclei with a pixel classifier instead. So I'm going through annotating what is nuclei, what is background, and then once I feel like this has been fine-tuned well enough, I can then ask the software to generate detections for each one of those nuclei. So each one of them will be a separate object. Yeah, sorry that this is really fast. We just have a lot of tools that we want to give you quick overviews of to show you what's out there. Okay, so I'm saving the classifier so I can reuse it on other images. And then I'm asking the software to generate detection objects of these nuclei, which I'm toggling on and off. I can then calculate measurements for these nuclear objects for all of the channels that are present within this image. And then again, once I have those calculated and added in, I can visualize those as I would like to help me get a sense of what might be going on with my data with a heat map kind of view. All right. So some quick pros and cons about using QuPath. So with a little bit of a learning curve, overall the the graphic user interface is pretty user-friendly. It has an incredibly responsive community backing. It also has that pyramidal file support, which I was mentioning earlier. You can easily set up batch scripting for analyzing lots and lots of images the same way at the same time. And it overall is pretty flexible and scalable as far as an analysis solution goes. Some of the cons are that it does have limited support for 3D images. So while you can open up 3D images in QuPath, you're not really able to analyze them in 3D the way that you might like to be able to. More complex analyses will likely require scripting knowledge, and QuPath's native language is Groovy. There are other languages that are supported though. And as of right now, the API documentation, so what the different commands are called in Groovy, is a little bit limited. So hopefully now that his team is growing, some of that might be expanded upon in the future. All right, so we're going to hand it over to Helen, who's going to talk to us about ImageJ.
Hello. So yes, my name is Helen, and Fiji/ImageJ is a open source image processing package. So Fiji is the batteries included version of ImageJ. If you've heard of one or the other, we recommend beginners just download Fiji because it has a lot of things pre-included in the download. So different plugins and functionality. So again, this is open source, free for everyone, and it is community developed. So there's been tons and tons of people working on these projects. It started originally in 1997 with Brain Raspberry and has grown exponentially from there. So pros, this is a tool that's widely used by many different fields. It's not just limited to, biological or cell analysis. It is community developed, so there's tons of different kinds of tools and resources that you can use for image processing. And it supports everything from the very basics of like contrast and color display all the way up through different machine learning functionalities. Some of the cons is probably that macro language. If you're looking for an easy way to get into coding, the macro scripting language is within Fiji cannot be supported by every plugin because it depends what the developers have written. Fiji can be somewhat slow with very large data. Virtual images can help with that, but you don't necessarily have all of the tool functionalities that you might like. And then documentation and plugin support may vary. So you might be trying to use a plugin that was developed 10 years ago that, may have some bugs or may not work as it could. So to get started, once you download it, you end up with this kind of toolbar where you have file, edit, image process, and then these like rectangle and line tools. That's the main functionality of like the Fiji window. And so in this, you have a lot of different like menu actions and like filters and different things that you can apply to your window. You have different tools for selection. I recommend just opening an image and seeing like kind of how much you can like quote unquote destroy it. Play with different filters, crop things, edit things, see what you can filter out and change. So this can be really great for looking at different types of pre-processing steps that you might want to use. And you can also right click or double click on different tools just to help you kind of learn what all the buttons do. If you get tired of going through all of the menu paths, you can use the search bar. So you can just press Control L to bring this up. So for example, if I want to run a threshold, I can just search for that. And then underneath the run button here, you have help option, which will link to any documentation on the website and the actual link to the source code. If you want to see like how a filter might be applied, you can like get the actual code for how it's working. And so what can Fiji do generally? So you can do things like profile plots, so estimating sizes of objects, looking at signal to background ratio. You look at histograms, different ways to display your image with brightness and contrast. Again, these filters are great for pre-processing things. So of course, you always want to make your measurements on your original data, but if you need to maybe do something to help get a better segmentation of something, you can use filters to do things like that. There's lots of different kinds of thresholds that you can run besides the default algorithm. And of course, you can get various measurements out of your images. So scripting and batch analysis is a great way to get into coding, or if you have, the same workflow that you need to run on like 500 images instead of just 5. You can just give it your folder. Fiji has these great template codes. So here we could just like add our code that we want to run into this and it'll process everything in that folder of the images that we have. And if you're new to coding, the macro recorder is a great way to help generate your scripts that you want. So here, there's an example where I'm just running a basic threshold to segment background and foreground, and then we're going to analyze the blobs that I have in this image. So I've opened the recorder using the search bar, and now everything that I click and type is going to be recorded as the script that you would use in the macro language. So I set a different type of threshold. You can see there's different code lines based on which settings I want to use. And then I quickly run analyze particles. Again, different settings will have different lines of script that you generate. But I can just then copy paste those two into a Fiji template. I would just add an open and save as statements, and I could run the same workflow on now, several hundred images instead of just the one, or manually going through one by one. So again, there's lots of plugins. This is community developed. So those are kind of the basic functions of Fiji. But if you know what kind of analysis you're going to need out of your image, whether it's segmentation, tracking, things like that, there's hundreds of different plugins that you can look through. There's a list on the website, as well as if you want to add them to your Fiji, you can do manage update sites in the updater window. So yeah, thank you very much.
Yes. Hi, everyone. Thanks for joining. And thank you, Helen, for giving an overview of imaging. I'm Suganya. I'll be talking about Cell Profiler today. Like other image analysis tools that you've been hearing so far, Cell Profiler is also free and open source software. It was initially developed by Ann Carpenter and Ray Jones, and it is being continuously developed by a group of software engineers and biologists. And most of the documentation of Cell Profiler is done by biologists so that the users are easy to follow documentation available. And the software is available to work on platforms like Windows, Mac, and Linux. And if you're a person who are processing a lot of images on the cloud, Cell Profiler can be run on cloud as well. To go into more details of how Cell Profiler works, okay, yes. This is very good for batch processing. You can load in all the images at once, and you basically create a pipeline in which you add different steps that you have to do on your images. And these steps are called as modules in Cell Profiler. Basically, you have modules for pre-processing your images or to identify the objects in the images that you have loaded, and also to process your objects that you have identified. And the main advantage of Cell Profiler is you can make a lot of measurements on the identified objects. And once you have made these measurements, you can export the data as either CSV or as a database to then do downstream analysis. To have a look at how the Cell Profiler interface looks, the download link has been provided in the resource document, and I recommend you to download the latest version. And once you've downloaded, this is how the basic interface looks like. On the left, you have the pipeline panel where you add in the different steps, which we call it as modules. And you can add in the modules by clicking on the plus button here, and this is the add modules. And once you click that, you have a pop-up of something like this image where the modules are categorized into different categories based on whether it is for image processing or object processing. Once you've identified the objects, then you can also do measurements. Once you add these modules on the pipeline panel, the corresponding settings appear on the right side, and like different parameters are also displayed. If you're not sure if any of the parameters, how it works, you always have these question marks all over the modules and different sections, and you can click it and it takes it to the documentation, and you have a very well documented manual where you have the the settings explained. Based on that, you can tweak the parameters and also choose the settings accordingly. For now, I'll be showing you a very basic demonstration of how to identify primary objects and secondary objects in monolayered culture cells, for which we'll be choosing these modules to be added into the pipeline panel. This is how you identify objects, and in Cell Profiler, you can choose which channels to be chosen to identify the objects. To choose here, I've given the DAPI to identify the nuclei, and for the secondary objects or the cell boundary identification, I've given the actin channel. Once you have identified the nuclei, there are different thresholding algorithms. If there might be over-segmentation or under-segmentation of the nuclei, you can tweak around the parameters to identify it properly. Primary object identification is very important in Cell Profiler because this will be used as a seed to identify your cell boundaries. And there are different ways to identify cell boundaries too, and you can choose based on your images. And you have both the nuclei and the cell boundaries displayed like this. And if you're satisfied with the object identification, then you can add in a lot of measurement modules. This is again similar. If you click on the plus button, then you have this add modules pop up. And when you go into the measurement category, you have all these measurements. For example, you can measure co-localization, or if you want to do intensity measurements or the distribution of intensity or size of the objects, all of these can be made. Although the list looks smaller here, you can literally make thousands of measurements because each of these modules will make a lot of measurements. And once you have added in the measurements, and of course, you can save this as a CSV or a database for downstream analysis. This is just an example image of a monolayered culture cells where you want to identify objects. But if you work with different images, there are different example pipelines that I've linked in the resource document as well. For example, if you want to identify objects from yeast patches, or if you want to do a speckle counting, that is also possible with Cell Profiler. And you can also do object tracking. And if you're a person who are working with deep learning tools like CellPose or machine learning tools like Elastic or imaging macros, this can also be integrated into Cell Profiler. Like you can use the pre-trained model from CellPose, or if you have custom trained models that can be imported into the Cell Profiler, and a lot of measurements that are available, or the modules that are available in Cell Profiler can be then used to integrate with your CellPose or the Elastic models. Similarly, if you have image scripts that you have already created to identify the objects, that can be integrated into Cell Profiler, and you can make a lot of measurements on those as well. You have a lot of plugins, and with the links of those plugins to access those plugins is available in the resource document as well. And to give a quick overview of the pros and cons, the main advantage is you can make a lot of measurements without having any programming language, and it is a tool that is very well suitable for high throughput imaging. If you do imaging from 96 well plate or 384 well plates, all of those images can be processed at the same time by creating a pipeline. So it is very well suited for batch processing. And you can also integrate the deep learning or machine learning tools like CellPose and Elastic with the plugins available. And the software is very well documented. And if you're stuck at some point, the help is always available on ImageSC, or the documentation is already well done. But if you want more help, please post your questions on ImageSC and people are there to help you. If you have a pipeline that is halfway through and if you want more help, please post it on ImageSC and that will be helped. And the cons, for now, if you're a person working with whole slide images or primitive images, for now, I think the support is not available, but it will be available in the future releases. And learning about input modules can be a bit tricky in the beginning, but we have a blog post and a video tutorial to help you with that as well. So I know this is a very basic demonstration of what I showed with the monolayered culture cells, a very elaborate video tutorial on beginner segmentation, and also more video tutorials are available for which the links are provided in the resource doc. With that, I'll hand it over to Tim to talk about Nabari. Thanks.
All right. Hi, everyone. I'll be talking to you about Nabari today, which at its core is really an n-dimensional image viewer, but which ecosystem is much bigger. I'm going to start with the pros and cons because it'll actually give you sort of the rationale for what I'm going to show you today. And one of the biggest pros of Nabari is that it's written in Python, and it's integrated very closely with Python, which can also be used for data analysis, meaning that you can keep your image analysis and your data analysis in the same place. I find the Nabari interface to be a simple and intuitive UI. There's a variety of image and data that is supported. There are now over 500 plugins, and the community is amazing. Nabari was released to the public in 2019, so it's the newest of all of these. And there are many different resources to be able to start using Nabari. But it's sort of an example for an early career working group. 3 years ago, I had no knowledge of Python, and I started building a Nabari plugin. Now, recently, I've been contributing to Nabari, and I'm a core developer. So you can learn to do these things yourself if you want. So for the cons, one of the biggest cons is that Nabari is Python. And the disadvantage of that is it can be hard to learn how to set up, even though there is a one-click bundle install, like something like Fiji would have. It also means that we sometimes break things. There's not really a direct path to success because Nabari is newer, so the documentation isn't as mature, and Nabari itself is experiencing some growing pains as we sort of shift strategies around now. Nabari itself is a simple interface. Here's a Tribolium beetle embryo. We have a 4-dimensional image. I was scrolling through Z, and now I'm scrolling through the time stack. We can change things like the color map or contrast very easily. One of the beautiful things about Nabari is that you have really great 3-dimensional interactivity. You could still scroll through time. Here it's showing you the labels that I've output from a processing pipeline, which all of the things that I'm showing you in this demo are available in a GitHub repository, so you could run them yourself. Here I'm showing you that you can annotate things with labels layers. It's very smooth and quick. You could also, for example, mask regions with our shapes layer. And you could similarly add things like points. Those things are still available in 2 and 3 dimensions. So viewing and annotating images is sort of the core of Nabari. But we also support a diverse set of image data. So here, for example, is a table-like data in X-ray format, and it's showing the surface temperature of the ocean over time. Not something we normally think about as image data, but there it is. Here is a texture of a coral on the left mapped to its three-dimensional space. Or on the right, we have a structural molecule of DNA, of course. It also supports other types of things, like here's vectors and points annotated on a tomograph image. And we also support fairly well large images. So here is remote viewing of an OOMIZAR file that is used with the Napari OOMIZAR plugin. And I pasted the internet link, and now I can zoom through the pyramidal layers and pan around. Not quite as good as QPath, but I think it still works very great considering all of the other viewing potential. So if you want to integrate Napari with Python, something like a script or a Jupyter notebook where you might want to do your data analysis, you can get results where on the left you can get tables of information about your data or plots, but then on the right in the same notebook you can visualize the results of your data. And so you have a one-stop shop for image analysis and data science. But let's say code is intimidating. Napari has many plugins available. This is the one that I started 3 years ago. It was very simple, but my goal was many things, but to partly bring batch processing to Napari. And in this, we're going to be using the Napari sample image of a kidney, and the magenta and green areas are glomeruli. So I'm labeling the background in red and my objects of interest in cyan. We have to do really sparse annotation. I'm annotating too much. And then in the plugin now, we can interact with the plugin, we can set a file, we can pick our feature set, what channels we might want to use for training our machine learning classifier, and then we can predict with that. The advantage of using a machine learning classifier for this is that instance or threshold-based segmentation would, of course, not really work well for these dual channel images. So we can view them in 2D and scroll through and see that the glomeruli are nice, or even in 3 dimensions, as this image is. But let's say you're kind of in that place in your work where you are scripting, but you want to share your work with colleagues who don't want to use code, and that's overwhelming. Napari makes it very easy to build widgets and plugins. So for example, let's say you've just had a function here which thresholds something according to the Otsu algorithm. And we just decorate it with Magic GUI and a few other what are called types in Python. And that is all you need in order to generate a widget, which means that, let's say in a core, you want to share some processing that you do with colleagues. We can also turn this into a plugin very easily with the guide from the Napari plugin template. So we can download different plugins, and here are just a few examples. Here's sort of a calculator-like image interface to do a lot of image processing. Here's deep learning with Stardust. Here is spatial omics data, or feature clustering, such as a UMAP of this different labels. So with that, Napari, in summary, is sort of the synthesis and meeting place. I think its strength is that it binds image analysis and data science very nicely together. Thank you.
All right, I will try to go through this pretty quickly. I realize we're right at the end here. All right, so this is kind of a where to go to get help. It's not actually specifically necessarily about help, because the first thing I'm going to discuss is Microscopy U. But basically, there's 4 or 5 things here, and I just want to go over. So Microscopy U is great for microscopy concepts if you want to learn about or are being introduced to microscopy like right now, or just want to look up something in particular in terms of a type of microscopy. Microform is great for microscope design, hardware, and a lot of sample preparation and type stuff. The ImageSC forum is great for image analysis, and also you'll see the microscope control software discussed earlier, Micromanager there. And the confocal listserv is a nice place for just all sorts of microscopy issues, events, jobs, and more. All right, so Microscopy U, again, good for a lot of the basics. If you just want to see all the different categories here that are covered, there are a lot of nice little interactive tutorials, and articles on everything from basic concepts like what is cover slip correction, why do you need a cover slip, how thick does it need to be, to more complex topics if you want to dig into like fret or storm, because you've heard of those, seen those in a paper, and want to learn a little bit more about how they work. The rest of the topics here, though, are, oh, sorry, it's nicely available online, unlike Pauly's Big Blue Bible. The microforum is on the interactive side. So if you want to ask questions about hardware acquisition specimens or theory, or kind of practical design choices if you're building a microscope or trying to set up some kind of wonky sample. Here's just some of the popular discussions about like designing open software for optical design, getting a microscope design built, how hardened media can compress your sample, information on live cell imaging, tips and tricks there, looking for a new color camera, like all these kind of topics are great for this particular forum. My particular favorite is the ImageSC forum. Been involved in this for a while. It has quite a few community partners now. The only requirement is an open source non-commercial compute license. Again, primarily image analysis, has a little bit of something for everything. There's the bulk of the posts, I would say, are new users asking questions. But there are quite a few expert discussions, which are just like in-depth analyses of a particular topic. We do have commercial vendors and plenty of discussions about commercial software. We just ask that there's no advertisements. Zeiss and Physiofarm in particular have members who are fairly active on the forum. And as you see, like some of the most commonly discussed topics are the things that we presented here today. You'll see a lot of guides of meta-analysis of techniques. This in particular was the analysis of InstantSeg, a new cell segmentation tool that's built into QPath in comparison with like CellPost, Artist, and some others. Again, the bulk of the topics are biological questions about how do I segment X type of cell or X type of tissue. But you'll also see posts on classification, import, export, scripting, all the little tricky things that can get you hung up when you're going through a pipeline. And it's not all just basic biological questions. You'll see questions like how to segment bees. There's a post on QPath for cheese with all the puns you might expect to find in something like that. Some of the in-depth discussions are things like color deconvolution implementations, best practices, and comparisons of tools for 3D dense ground truth annotations. It's nice that people have spent the time to post their experiences and stuff on these so you can maybe just jump straight to a particular tool. It is also very commonly used. You don't need to actually sign up for the ImageSC forum to get access to all the things that people have discussed. And you see that there are some topics that just have a lot of views. And it is interactive. Like there are some posts that have gotten hundreds of responses. I was especially involved in the designing a QPath workstation discussion. So it is active. You don't need to usually feel like you're shouting into the void. Okay, last one is the confocal listserv. This is a little bit less accessible. It's just signed up emailing list, but they do have a amazing collection of very experienced microscopists discussing everything that you might want to know about microscopy. I've posted on here a few times. The only real downside to this is that it is not a forum, so it's not quite as easy to search. If you are interested in a microscopy career, I'd recommend signing up for this and just start collecting the emails in your email client so you can more easily search them. Otherwise, it does require like a sign-in to search through old emails. If you want to find it, just search for confocal listserv. You'll find something from the University of Minnesota Twin Cities confocal microscopy list. And if you want to sign up, you have to go like into this kind of page here, find the hamburger, and either subscribe or unsubscribe. And in general, just advice for everyone asking questions out there. Most of the people on most of these resources are helping you for free, and they can't see what you see. You're looking at a screen, you know what your experiment is, you know what features in the image you are actually looking for to segment or whatever. So providing images, the text of any error messages, not just pictures of the error message, what operating system you're using, what version of the software you're using, and any code that you're actually running would all be very helpful for the people who are trying to help you. So, include some information in there, not just, I can't get my cells to segment. And yeah, I see Lisa's already started here, but if you have any alternate help options that you can provide, I'd love to see everyone throw those into chat right now, and we can add them onto our resources document. And thank you all. I don't know if anyone else has any last comments you want to say, or can open it up now for questions. And Kristen, you can take back control.
Right. Thanks. Sorry, I have a slightly loud conversation occurring next to me, but thank you guys all so much for coming. And we really appreciate you all taking the time. We have our QR codes up here for link to the survey again, the resource document that we made for you guys on everything we covered today, as well as some alternatives to some of the things that we discussed, and the image forum post. So we are going to, yes, you can absolutely share your resource list with whoever you want. Yeah, please share. This is recorded, so it will be available on YouTube at some point, and I'm sure some emails will go out when that's available. And yeah, we will try to migrate all of the questions that we get and answer them in a reasonable amount of time at the image forum post. So just kind of keep an eye out on that. You can join the image forum and bookmark it, so you will get that post, so you'll get notified as things get added in. You can also add questions there if you'd like. So with that, we'll conclude today's meeting. For the people who were helping with presenting, if you could hang back for just a couple moments, that'd be great. But other than that, thank you guys so much. Appreciate you being here.